So Paul, I'll go ahead and pass it over to you.
Thank you. I'm very excited to welcome Dr. Rob Cramer here tonight. I had the opportunity to hear him speak this past spring at a Dartmouth seminar, and was just fascinated by his insights into how fungi, at least certain species of fungi, are changing under environmental pressures from climate change and how that's impacting their pathogenesis in humans. Dr. Cramer first became interested in the genetics of pathogenic versus non-pathogenic fungi as a grad student at Colorado State University, and then at Duke University Medical Center worked quite a bit on Aspergillus fumigatus and how it can be in certain situations more pathogenic in humans. He's now a professor of fungal pathogenesis at Dartmouth's Geisel School of Medicine, their Cramer laboratory. And yeah, let's jump right in and hear what he has to say. Thanks, Rob.
Yeah, thank you, Paul and Emily, for the kind invitation to talk to you tonight about my favorite group of organisms, the fungi. I think, my two goals tonight for the audience are, to kind of just educate you a little bit about the fungi as pathogens. I'm sure most of you on the call tonight didn't wake up this morning worried about getting a fungal infection that might be life-threatening. But the unfortunate news that I have for you tonight in the context of climate change is that, potentially that may be a concern moving forward in the future. And we need to be proactive with our research and environmental monitoring to prevent the increasing incidence of these infections. So the first part of the talk, I'm going to talk a little bit about fungi, just kind of introduce you to this group of my favorite organisms. And then the second part, we'll really kind of dive into how the pathogenesis and incidence of disease associated with these organisms is actually quite rapidly changing, not only globally, but also in our backyard here in New England. So let's go ahead and get started. It's my dinner time, and so I'm just gonna torture myself by looking at this awesome pizza. So, I say that tongue in cheek, but the mushroom topping on this pizza illustrates a very important point and kind of a theme that goes throughout the talk, and why the infections caused by fungi are incredibly difficult to treat, and why their increased incidence is a major concern. Is that fungi and humans are very closely genetically related. In fact, if we go and look at the tree of life, so over in the right corner here, if you can see my pointer, fungi actually diverged from the animals about 1.5 billion years ago. And we can make that estimate based on the molecular clock theory and looking at the DNA of fungi in animals. And it's a really, robust prediction based on a lot of data. And what's interesting phenotypically, how these organisms differ, I mean, obviously we look different, completely different, but metabolically we're very different. So, for example, the fungi are able to synthesize all their own amino acids where we have to acquire them from the environment. And so the fungi maintain those genes over millions and billions of years, where we lost some of those genes during the course of our evolutionary history. And that has implications for how we treat these infections and deal with these organisms in the clinic. And so really fascinatingly, in the last few years, we've actually now have fossil evidence to support the molecular clock data. So this was from just a few years ago up in the Arctic, that the world's oldest known fungal fossil was discovered. And you can see a very close image of it here. Now, there is a little bit of controversy of whether or not this is a fungus, but a lot of the data associated with this fossil is increasingly supporting the hypothesis that the structure you're seeing here is a fungus, including some really high-tech microscopy being done, looking at the composition of this material, which suggests it contains a molecule called chitin, which is a polysaccharide that's found in the cell wall of these organisms. And so really good convergence of molecular biology data with the fossil record, showing us that fungi have been on this planet for a very long time. They've actually potentially even predated the arrival of plants on land, which is fascinating because it brings into question, well, what were these fungi feeding on, and what were they using as an energy source when they were growing in the climate 1.5 billion years ago? So we're going to move up in time here and talk a little bit about dinosaurs. Hopefully you were expecting that tonight from a lecture on fungi. But of course, the dinosaurs experienced one of the biggest changes to our climate that has been recorded in the fossil record. And that happened due to the meteorite that struck our planet 65 million years ago. And so, I think a couple points here I wanted to make is that this event caused massive climate change. And so it caused the mass loss of plant life throughout the planet for multiple reasons, from all the debris, from the increased volcanic eruptions, which block sunlight. We have wonderful fossil record evidence of this. And at the same time, what that caused is a change in the climate. So temperatures cooled, the planet became quite moist. Obviously, the dinosaurs went extinct, but that raised an interesting question: is why did the dinosaurs go extinct? And why didn't they just come back? And why did all of them die? And so the fossil record actually has really good evidence that changing climate promoted a massive proliferation of fungi. And what's interesting there is that those fungi then were growing predominantly at temperatures that were quite cool. So, ambient temperatures between, 10 and 25 degrees Celsius, you can see in the graph in the upper right, is where the vast majority of fungi like to grow. And if you go out on the curve looking at the x-axis here and you get over to mammalian body temperatures, you can see that very few fungi actually grow at mammalian body temperatures. And so the hypothesis that was put forward by one of my colleagues, Dr. Arturo Casadevall, was that this created an enormous selection pressure that allowed the mammalian endothermy to arise. And so mammalian endothermy and homothermy really creates this thermal exclusionary zone that protects mammals against invasive fungal infections. And so this is a major reason why when you woke up this morning, you weren't worried about getting a systemic fungal infection, is simply the fact that you are an endotherm and have high body temperature, and the vast majority of the fungi in the environment cannot grow at these temperatures. And so we'll talk a little bit more about this later in the talk, but obviously this has relevance for our changing climate now, where we're seeing a warming of the environment, and also importantly, increased periods of time of very high temperatures that potentially could be a selective pressure for promoting the adaptation of these fungi that are growing at 30 degrees Celsius, that potentially very quickly can adapt and begin to grow at human body temperature. And so a lot of these species, which previously we didn't worry about as pathogens, may be able to break this thermal exclusionary zone in the near future. And there's laboratory data to support this. These organisms rapidly adapt to changes in temperatures. It's really quite remarkable how quickly they can adapt to growing at higher temperatures. So, when we talk about the fungi, we're talking about eukaryotes. Again, those mushrooms on your pizza, you are genetically related to them. You have a lot of the same genes and proteins that fungi do. So most fungi that you're familiar with most likely are multicellular, just like us. The exception are the yeasts. And so yeasts are unicellular fungi. Maybe some of you tonight are having a beer or a glass of wine while you listen to the talk. And so that's courtesy of the fungi, those unicellular yeasts. The molds or filamentous fungi are our multicellular fungi, and they have these slender filaments, which are called hyphae, which perhaps you saw in the image of the fossil that I showed you previously. And those hyphae can interact with each other and form what's called a mycelium. And the mycelium is what you see when you see a mushroom in the forest. So that is a mycelium when you see that mushroom. So they grow by obtaining nutrients from the environment, and they secrete enzymes to do this. And so if you think about pathogenesis and these organisms causing disease, they essentially break down your tissue. They secrete enzymes that can degrade proteins and polysaccharides in your body, and then they absorb that material. And so fungi are critical members of the environment because they are involved in recycling carbon and nitrogen. So all that plant material that died when the meteorite hit, the reason it was able to be digested and then further support life was because of the fungi. And so that provided a really rich fertile ground for fungal expansion. So as I mentioned, the fungi contain these really unique structures called cell walls that protects their cells from the external environment. Chitin is one of the polysaccharides that's in the fungal cell wall. the cell wall is important in interacting with hosts. It's really the first part of the fungus that your immune system sees. Your immune system really reacts quite strongly to fungal material. So that's why we have diseases associated, like asthma and allergy associated with fungi. A lot of it has to do with these fungal polysaccharides found in their cell wall. And these cell walls are really rich storage reservoirs of carbon. So there's actually quite a bit of research going on utilizing fungi. Can we use fungi as a carbon sink in the environment to sequester carbon? Fungi, their membranes contain ergosterol, and I bring this up because it's the fungal equivalent of cholesterol, and the biosynthesis of this molecule is almost the same as the biosynthesis of cholesterol in your body, and so the antifungals that we primarily use in the clinic target this molecule, ergosterol, and it is really important in temperature adaptation to these organisms because cholesterol, ergosterol, they're really important in controlling membrane fluidity, which is directly impacted by temperature. So we're really interested in this molecule and how it's potentially going to change as the climate warms. There's these really cool fungi. Many of them are pathogen that are dimorphic. So they actually can switch between being a yeast and a hyphal form. And we're going to talk about one of these that's becoming important here in New England due to climate change in a few minutes. So we've talked about fungi as pathogens a little bit. We've talked about them as being saprophytes, recycling carbon and nitrogen in the environment. And so I hope that illustrates this point that these organisms are incredibly flexible in how they live and grow. And so, there's, as I mentioned, they're saprophytes or saprobes, they're parasites, but they're also mutualists. And we really don't have a lot of time tonight to get into this because we're going to focus on human disease. But the fungi in the soil, these organisms called mycorrhizae, most of the plants that you see growing, the trees, the grasses, all have a fungus associated with them. And that diversity of plant life that you see is primarily due to this interactions with these mycorrhizal fungi. And how climate change is going to impact these mutualistic interactions is it's a very important area in research and one that people are actively exploring. Another little quick note is lichens are a mutualistic interaction between fungus and algae. Some evidence to suggest that perhaps those interactions might not always be mutualistic, but those lichens you see all throughout New England, there's a fungus in there as well. Okay, general life cycle. This is important because this is where the infectious propagules come from. And so many fungi produce asexually. So you have a spore, it lands in an environment that it's conducive to growth. It germinates either into a yeast or hyphae, which then form the mycelium. And that mycelium structure can then produce the spore-producing structures. And this is really common. They produce these in massive quantities. In New England here, many of us have mold in our homes. Perhaps you've seen the colored spores on your wall. You've probably at least seen them on your bread if you leave your bread out too much. And those spores are coming from asexual reproduction. The mushrooms you see are actually sexual reproduction. So the mushroom actually is a fascinating structure. So it's actually two different fungi, same species, but two different mating types that have fused together. And that dikaryotic stage where you have nuclei from each of the different sexual partners living together in the same cell is what produces the mushroom. And eventually those nuclei fuse within those mushroom and they undergo meiosis and you get sexual recombination, just like you do in mammals. And that produces genetic diversity. And then those mushrooms, as they die and they go away, then they release their spores into the environment, and those genetically diverse spores can now colonize new environments. So here's just some pictures of some yeasts, including a picture of a blood agar plate. If there's any clinical microbiologists on the call, they obviously recognize the blood agar plate. These cells are much bigger than bacteria cells. These yeast cells here are at least 5 to 7 microns in size. Most of the yeast produced by budding, but there's other mechanisms that they can reproduce as well. So this is the organism again that gives us beer, bread, and wine, Saccharomyces cerevisiae. Here's the molds. You can see the asexual reproductive structure here of this mold here. You get these beautiful structures that form that produce the spore chains that release the spores. The hyphae grow into substrates out in the environment. Unfortunately, in humans, they grow into your tissue. We generally don't see asexual reproduction in the human body when you have an infection. The fungus stays in this filamentous form. And, as I mentioned, on your walls or on bread, this is often what you've seen. This is a colony, a mycelium or a biofilm of fungus. The coloration comes from the spores. It's a form of melanin, similar to the melanin that's in your skin. And it protects those spores from UV radiation, from heat stress, oxidative stress. It's a known virulence factor of fungi. And again, here's a picture of the mushroom just to show you that. Again, the hyphae are in the soil. When conditions are right, two different mating types come together and fuse, and that's what forms the sexual reproductive structure, which is the fungus. Mycology has all kinds of great terms. The vocabulary is pretty intense. So the mushroom itself is called the basidiocarp for the aficionados in the audience. And then spores are released underneath the gills of the mushroom. That's where the spores are held. So fungi are throughout our environment. They've had a huge impact on humanity. They do all kinds of things that we benefit from every day. I mentioned the mycorrhizal associations. They're important biological control agents. This is probably going to increase as we see increased changes in the geographic distribution of insect pathogens like mosquitoes, for example. They're very commonly now being used as a food source. There's several new biotech companies that are using fungi to generate protein products. Some of them look just like a chicken breast, and I can't say they taste just like chicken, but they don't taste that bad, actually. So you're going to see more and more of that in the future. Many of the enzymes that fungi made are used to make cheese. A climate change related story there that's not good news is these beautiful blue cheeses that come from France. Because of the changing climate, the caves that these cheeses are incubated in that allows them to acquire these fungal communities that give these cheeses their amazing flavor. We're starting to see a reduction in those species, in those caves, due to warming of the caves and changing in the environment in these caves. So if you're interested in that, there's quite a bit of literature developing on that. Kind of a scary thing for cheese lovers. Antibiotics are a big one. A lot of antibiotics come from fungi. We're going to talk about a couple here in a minute. And those antibiotics are produced by fungi to interact with microorganisms in the environment. And again, climate change is having a big impact there, and how these organisms are interacting out in the soil as the soil changes in response to heat stress, carbon stress, changes in pH, micronutrient availability, and that's going to change the spectrum of antibiotics that these organisms ultimately produce. We're mostly going to talk tonight about the bad. Unfortunately, we're going to talk about fungi as human and causes of human and animal mycoses. We're not going to get too much into allergy, but we'll talk briefly about it. And we'll also briefly talk about plant diseases, though we don't have too much time to talk about that. So we're in New England here, so I thought I'd show a slide with the Salem witchcraft trials. You might be thinking, well, how the heck does this have anything to do with fungus and climate change? And it actually is directly related to climate change and fungi. And so there's now excellent evidence that in the 16th, 15th, and 14th centuries, when the witchcraft trials were at their peak, particularly in Europe, the cause behind this, there's a lot of causes, but one of the actual practical causes actually has to do with a fungal infection of plants. And so in years where the climate was moist and wet, the wheat, which was the primary food source particularly for the wealthy at that time period, was unable to grow. It would just get wiped out by different fungal infections. And some of the lesser desirable grains like rye and barley were able to grow, but they also were able to get infected by a fungus called Claviceps purpurae. And so the rich would be able to, and the wealthy would get access to the wheat that was able to grow, and then the peasantry was forced to eat the rye and the wheat. But unfortunately, the rye and wheat was infected with this fungus, and you can see these little structures here, these ergots. It's named after the little spur on the back of a rooster's leg. Don't ask me why. It does look just like that, so that's why it was named. But these little ergots are essentially masses of fungus, and that mass of fungus produces an antibiotic, which happens to be a hallucinogenic alkaloid. And so it's essentially the precursor to LCD. And so when the peasants would eat this, it would cause two different types of gangrene, one of which would restrict blood flow to the extremities. So you get gangrenous ergotism. And so this would cause arms to turn black, which was one of the signs that a person was a witch. And then the second lesser side effect was convulsive ergotism, where the people were essentially high on LSD and they would lose control of their body, and then they would be called a witch. And so, again, here's a link between a plant disease caused by climate change impacting human society on a very grand scale. Everyone knows about penicillium. It came from a fungus. Again, the spectrum of antibiotics that these organisms are going to make, may change, perhaps for the better. Perhaps we'll discover some new antibiotics due to changes in the climate. But just one example here of how these organisms have directly impacted human well-being. One of the greatest discoveries of the 20th century. Certainly changed the outcome of World War II. And then more recently, economically especially, one of the most successful drugs economically of all time are the statins. It's produced by a fungus in my favorite genus, Aspergillus. And Goldstein and Brown won the Nobel Prize for discovering, how cholesterol metabolism is regulated. And then the statins, which inhibit HMG-CoA reductase, was one of the first steps in ergosterol and cholesterol biosynthesis, both in fungi and in animals, are targeted by the statins. Okay, so now let's talk about fungi as pathogens, now that we know a little bit more about them. So surprisingly, it's only been recently that the World Health Organization has recognized fungi as important pathogens on the planet. And there's a lot of reasons for that. It's really quite goes back to, if you look back through the history of medicine, you see very few references to fungi and human disease until about the 1960s. And so in the 1960s, we started to see infections caused by fungi coming up in the clinic more commonly. And then from the 1960s all the way to the 2000s, there's just been this steady increase in the incidence of these infections. And so the rapid advances in medical therapies for autoimmune diseases, for cancer, they often are using molecules that target the immune system. And so essentially what's happened is we've seen a change in the patient populations that are immunocompromised. And that's provided an opportunity for these organisms to come in and cause lethal disease. And so the World Health Organization in 2022 came up with a white paper. These are the 4 organisms that are at the top of the list, so to speak, based on their abundance and causing human disease. Cryptococcus neoformans, it's a yeast. Candida auris, which we're going to talk about tonight. Candida albicans, and then my favorite, the organism I work on in my laboratory, Aspergillus fumigatus, is the mold. And so, as I mentioned, why is this recent? Well, some of it has to do with the advances in modern medicine. So in 1976, we discovered a antibiotic produced by fungi that actually is immune suppressive called cyclosporin, which made organ transplantation a reality. So in order to undergo an organ transplant, you need to suppress the host's immune system, and that makes them susceptible to fungal infections. In the 1980s, we had the HIV pandemic begin. And then rapidly, the number of immunocompromised patients rapidly increased. Many of these patients would ultimately succumb to fungal infections due to a massive loss of function of their immune system. That continues to this day throughout the world. Fortunately, in the developed world, we do have highly active antiretroviral therapy, but there are parts of the world where that therapy isn't readily accessible. And patients that have AIDS do still succumb to fungal infections. And so Cryptococcus neoformans, which I mentioned, is the number 2 cause of infectious mortality in Africa. And it's due to uncontrolled HIV infection in that continent. So in the 1990s and 2000s, we had this rapid proliferation of immunomodulatory therapies. That continues to this day. Remarkable advances in the treatment of cancer and autoimmune diseases. And often those advances get thwarted by these fungi. Very difficult to diagnose and treat infections in these patients. You know, recently with the COVID-19 pandemic, as well as influenza pandemics, we now know that viral infection causes susceptibility to invasive fungal infections. And so there's a lot of literature now trying to understand how these viral infections, which are increasing, cause susceptibility to fungal infections. And we've done a little bit of work in that area over the last few years. So the big question now, though, is, you know, how will climate change contribute to this increase in diseases caused by fungi? And, you know, we're really at the early stages here in answering this question, and it's going to take a concerted and focused research effort to address this question. But we do have some data, and we're going to talk about that tonight. So some of the potential impacts of climate change on human fungal pathogens are going to be alterations in geographic range. So we're going to talk about this tonight. I'm going to show you examples now, including one here in New England, where fungi that previously were found in a very specific part of the world are now being found in other parts of the world. Okay. Increased dispersal events. So again, really good data showing that fungi are having massive dispersal events caused often by increases in traumatic weather events, but also the changing climate. Fungi, that asexual, those life cycles I showed you, they're very intimately tied to the environmental conditions. And so as temperatures warm, as the environment becomes either more or less moist, depending on the part of the globe you're in, we're getting changes in the production of those infectious propagules, those spores. And so that's increasing their burden in the environment. We're going to see changes in susceptible host populations. So we talked about iatrogenic causes, you know, coming from the clinic, but the environment is going to change. We're going to see climate refugees moving from one part of the globe to the other where they don't have immunity to the local fungi. We're going to see individuals moving or living in environmental conditions that causes an increase in their susceptibility to fungi because of perturbations to their immune system. Going to show you a couple examples tonight of trauma from extreme weather events. The number of extreme weather events is dramatically increased in the last several years, and several of these have been associated with outbreaks of fungal infection. One of the areas that I'm most interested in is pathogenicity and virulence factor emergence. So the environmental pressures that these organisms are under, their biology is going to change in response to climate change, and those changes in their biology are going to influence how they interact with us as hosts and how they are able to progress disease or cause disease in different host populations. And we'll talk about one example that's emerged in the last decade. As the fungal biology changes in response to temperature, we talked about ergosterol, that molecule in the membrane. It's really important for the organism's ability to thrive at high temperatures. And it unfortunately is also the target of our antifungals. And so we're seeing increased drug-resistant isolates in the environment, in part due to changes in fungal biology, but also in part to what I mentioned earlier, the increase in plant diseases caused by fungi, which is causing the agricultural industry to use more antifungals in the agricultural industry, which is driving resistance in the human pathogen population that then is manifesting in the clinic. We'll talk a little bit about that in a few minutes. We don't have time to talk too much about this tonight. You know, changes in vectors. There's not many fungal diseases that are vector mediated, but there are some. So the most common fungal infections are dermatophytic infections. So infections of your skin, athlete's foot, jock itch, also mucosal infections like vaginal yeast infections. These can be vector driven, and the changes in these vectors as they change with the changing environments is going to impact the incidence of and the geographic distribution of these dermatophytic and mucosal fungal infections. So the consequences here are quite severe. So we're going to see increases in human disease that includes both systemic infection as well as allergy. Disease outcomes are going to worsen with increasing drug-resistant isolates. There is the high likelihood of animal extinction events. We don't have time to talk about those tonight, but fungi are driving the extinction of many amphibian species across the planet right now, in part due to climate change. Our livestock health is going to decrease as livestock become more susceptible to these infections. As I mentioned, plant disease is a huge problem, and so food security and abundance is going to be important to monitor because nutritional immunity is huge. So one of the risk factors for increased susceptibility to fungal infections is poor nutrition. And so if our food supplies are affected, we're going to have an increased population that has nutrient deficiencies that's going to result in susceptibility to multiple types of infectious disease. And economic disruption has the potential to be quite severe. So here, I'm just going to give you some vignettes, then we'll wrap up tonight with each one of these impacts. So here's a local one from Vermont. This surprised a lot of us in the field. Recently, just came out, the CDC published a report in 2024 in February that this fungal infection called blastomycosis has, increased dramatically in Vermont. And so the state where this infection has primarily been associated with was Wisconsin. You can see the incidence in Wisconsin of blastomycosis was around 2.1 cases per 100,000 people. Well, what this report found was that in Vermont, we're up to 1.8 cases per 100,000 people in Vermont. And so there's been over 114 individuals impacted, in the last few years, many of whom succumbed to the infection. And this illustrates a huge point, this change in distribution of organisms. So there's a whole group of fungi which we have traditionally called the endemic mycoses. So these are fungi that are only found or were only found in specific geographic regions of the world. And so blastomycosis fell into this group of fungi. And so now what we're finding is we're finding these fungi in places we've never seen them before. And now, it's been estimated now that 10% of illnesses caused by fungal infections are now being diagnosed outside regions where these fungi are known to be endemic. Another one I don't have time to talk to you about is coccidioides mycosis, which previously was pretty much restricted to the desert Southwest and certain parts of South America, but we're now finding coccidioides mycosis up in Washington, in the Northwest. It's pretty unbelievable, actually, how quickly the range of these organisms is expanding in the last few years. So what is blastomycosis? It's one of these dimorphic fungi that I mentioned. So out in the environment at ambient temperatures, it grows as a mold. But when you inhale a particle, at human body temperature, it turns into a yeast. And this is, you can see it, and this is in human tissue right here, this yeast, very distinctive yeast with this mark where it reproduces that distinguishes it from other yeasts. That's probably too much detail. The infection occurs through inhalation. And so, what's probably happening is we've had a lot milder winters in the Northeast here. The organism is probably thriving in a warmer climate now in New England. And so we're seeing an increased burden of the organism in the environment, coupled now with an increased exposure to individuals here in Vermont and New England. So here's the life cycle. Again, this is from the CDC's website now. It's out in the environment. It's a saprophyte growing in warm, moist areas, recycling carbon and nitrogen. But now the endemic area has changed dramatically. It used to be just the upper Midwest, and now it's all the way down in the Southeast, up into New England here, which is quite surprising to a lot of us. Okay, so that was one example. There's other examples. We don't have time to talk to you about changes in geographic distribution, but I'm happy to answer any questions there later tonight. So increased dispersal events. As I mentioned, these airborne dispersal events, the fungi, the propagules, the spores, the conidia, this is kind of what they're made for. They're made to be dispersed through air currents. So we're expecting an increase in the environmental burden. That's because warmer temperatures are creating more air turbulence. I think anybody that's flown in the last couple of years has noticed that, wow, there's almost always turbulence on my flights now. That's in part due to these warmer temperatures. And so this is allowing more horizontal and vertical transmission of fungal spores. As I mentioned, the warmer temperatures are altering spore release patterns and the species prevalence in a given environment. It's really fascinating biology there that we don't have time to talk about tonight. We do know the Canidian spore counts are increasing of these organisms. And so as the temperature increases, they tend to produce more spores. And these more spores have been shown in many studies. The amount of spores in the air in hospitals have been shown to directly correlate it with an increased incidence of human disease, particularly caused by mold species like Aspergillus. And so, somewhat obvious, these traumatic weather events, tornadoes, hurricanes, can release large amount of infectious propagules in a very short period of time. And this can be associated with not only changes in incidence of disease, but also contribute to these changes in geographic distribution of these organisms that we're witnessing. Okay, changes in host susceptibility. People themselves are going to change and become more susceptible to these infections. We talked about climate refugees with no immunity to these endemic fungi. Climate has impacts. We know that increased UV exposure alters your T cell responses. It inhibits complement activation. It causes production of immune signaling molecules called cytokines that are not good, not the ones we want for controlling a fungal infection. And so, as the UV exposure increases, we're going to see alterations in human immunity. This is an interesting, I think some of you probably know this, some of you don't. Our human body temperature is decreasing. It's been decreasing about 0.3 degrees Celsius per birth decade. And so what does that mean? Well, we're getting a narrowing of the thermal barrier with those fungi, right? We're slightly lowering our body temperature, but that's all it takes to introduce a new group of fungal species that might be capable of causing disease in people. Your circadian rhythms are intimately tied with your immune system function. As the climate changes, we're going to see disruptions to our circadian rhythms, which will alter our immunity. And I mentioned the nutritional immunity already. It's an important component of our immune system. And as we have increased pathogenesis of plants, reduced loss of food, loss of nutrition, we're going to see changes in susceptibility to these infections. And here's just one example of that. This is a fungal pathogen of plants called stripe rust. The species is Puccinia striformis. So what you're seeing, the data here you're seeing is pre-2000. These are isolates of the fungus that were collected. And you're looking at the numbers are days. So this is days that it took the fungus to infect the plant, essentially. And so at 12 degrees Celsius, pre-2000, about 10 days on average. And then at 18 degrees Celsius, there was no change. So it still took them about 10 days, even maybe a little bit longer. So the pre-2000 isolates didn't like the higher temperature. But post-2000, as we collect this species, we see a change. No real change at the colder temperature and how many days it takes to infect the plant. But wow, look at this. 18 degrees Celsius, you're seeing a 3 to 4-fold decrease in the number of days it required for this organism to infect the plant. That's a massive change and is having huge implications for wheat crops across the world, particularly down in Australia right now, where this organism is causing a lot of damage to wheat crops. And so very short period of time, you see how this organism is adapted to cause more disease at higher temperatures. Okay. Trauma events. So tornadoes, volcanic eruptions, flooding, hurricanes, forest fires, all these are increasing with climate change. They're changing the levels of moisture and humidity, which is altering fungal proliferation. They're damaging our housing, increased moisture, massive fungal growth after flooding events. This contributes to allergy, asthma, poor air quality, poor structural quality in our housing. Fires cause massive increases in spore dispersal. It can activate spore germination. It increases housing loss, which increases exposure to outdoor molds and organisms. The flooding is increasing the waterborne fungi that are being brought to land. And if the right environment happens, these waterborne fungi can cause human disease as well. These events are altering soil chemistry, which is altering the abundance of certain fungi in specific soil environments. And then traumatic injuries. So, from tornadoes, from hurricanes, there's actually quite a bit of traumatic injuries that happen, which can breach our innate immunity and cause infection from organisms we previously didn't consider pathogens. One of the most recent examples was from the Joplin tornadoes a few years ago, where 14 individuals died, not from the tornado, but from traumatic wound injuries from the tornado. You're seeing the path of the tornado here. each one of these dots was a case, an infection by this fungus right here. It's a Mucorales fungus. It's not normally associated with human disease, but because these individuals had blunt force trauma from the tornado, that organism was introduced into their bloodstream and caused systemic infections, killing quite a few of them. So as we have more of these types of events, we're seeing more of these climate-associated traumatic injury-related fungal infections. Okay, here's a big one. This has kind of been the canary in the coal mine for a lot of us in our field. So the emergence of new pathogens due to changes in pathogenicity or virulence. This one's been all over the news. Here's an example from the New York Times, a mysterious infection spanning the globe in a climate of secrecy. This is Candida auris. So it's a yeast. It happens to be drug resistant. 90% of the isolates are at least resistant to one antifungal. Scarily, 30% of the isolates are resistant to two. This one it really is a remarkable story. So as I mean, the canary in the coal mine with climate change and emergence of new pathogens. So there is no record of the existence of this organism in the clinic, in the hospital setting prior to 2007. There's been a lot of bank samples, a lot of isolates collected for years, dating back to the 1960s. Not a single isolate of Candida auris was collected from a hospital setting, from a human being, prior to 2007. But then all of a sudden, simultaneously, on 3 different continents, 3 distinct phylogenetic clades of this organism emerged in patients. It's been remarkable. Here's the map in 2018. Here's the map in 2021. The organism has spread throughout the globe. I think we're up to 6 different clades. And so what that means is you have 6 independent emergences of this organism. These organisms between clades have, genetic, mutations in the hundreds of thousands, and yet the organism is finding its way into all parts of the world. And so what is going on here? And the answer is we don't know. We don't know where this organism came from. We don't know why it's suddenly a human pathogen. We have some recent data going back here that we've been able to finally find some environmental isolates. And those environmental isolates that were isolated from the environment, from the ocean, from an ocean environment, are less thermotolerant than the clinical isolates. So there's been a hypothesis put forth that's being tested, is that this is an organism that has emerged due to changing climate. So the global warming is responsible for raising the ambient temperatures, and this has driven changes in Candida auris populations, as it's now previously was associated with plants. It's gained a little bit more thermotolerance and a little bit more tolerance to salt. And so now it's being picked up by birds in these wetland environments, and the birds then distribute it to rural environments where you get interactions with farmers and livestock. And then eventually it's made its way into the urban environment, where it's spread rapidly throughout the medical care system across the globe. So again, this is a hypothesis, but the simultaneous emergence of multiple independent clades of this organism is really striking, as well as its thermotolerance, and it's really a striking story here of how relatively small changes in the climate can potentially select for a new pathogen. So another change of the climate is oxygen. So climate change and fossil fuel use is driving decreased oxygen levels, and this can change the virulence and pathogenicity of organisms. This is what my lab studies. So this is a little vignette from my lab. So ocean models right now are predicting a decline in the amount of dissolved oxygen available in the global ocean by 1 to 7% by the year 2100. And so this is really important because fungi, like you and I, they require oxygen for their biology. And so, the fungus I work on is the mold Aspergillus fumigatus. And so you inhale the spores into your lung, they germinate in the airway. And once they germinate into those hyphae, they eventually form these biofilm-like structures in your lung, which cause tissue damage as the organism secretes enzymes In order to form these biofilms, it actually requires oxygen, but to maintain them, it has to be able to adapt to a low oxygen environment. And so as the organism, as these spores germinate and forms these multicellular communities, you can see all these filaments interacting with each other, forming these beautiful structures that unfortunately are able to cause disease. These structures have these really complex metabolic gradients within them, including a gradient of oxygen. And so depending on where the cells are within these biofilms and where they are within your tissue, when they're highly oxygenated, they're susceptible to antifungal drugs, but when oxygen levels are reduced, they become more resistant. And so we're trying to understand the mechanisms through which oxygen impacts drug resistance and biofilm formation with Aspergillus. And so here's some data from our lab showing you that low oxygen is really important for being able to maintain a biofilm. So if you take an Aspergillus and make a mutant of it that can no longer adapt to a low oxygen environment, that's this strain over here, this SRBA strain, it can't form these biofilms anymore, and it can't cause disease. But if you now take that strain, or a strain that doesn't like low oxygen anymore, and you start to change its environment, you start to take away the oxygen slowly, like what's happening out in the environment now, the organism has time to adapt. It has time to evolve. And so if you do that in the laboratory with that strain, what happens is that long-term growth in these low oxygen environments restores its ability to form these biofilms, and then it is able to now promote more rapid disease progression. And so, here we have a strain that grew okay in a low oxygen environment, but after living in a low oxygen environment for a long time, you can see that the colony now looks better. It looks more happy. There's more biomass there, all that white tissue. And you can see that quantified here. It's able to make more fungus in these low oxygen environments. And then if you go into animal models with these strains now, they progress disease much more rapidly than the strains that didn't grow and evolve in these low oxygen environments. And so again, as the climate changes, these micronutrients, we're going to start to select for fungi that are able to deal with that, and that the convergence then of environmental adaptation and adaptation to a human host, unfortunately, is very, very tight. Okay, I mentioned drug resistance. So there's only 3 antifungals that are available to treat these infections. 3, that's it. And 2 of them target ergosterol. And so this makes our ability to treat these infections already limited, irrespective of climate change. But as I mentioned, climate change is causing an increased use of azoles in agriculture. And so what's happening is that this azole usage in the environment is driving the emergence of azole-resistant fungal strains, including those of the organism I study, Aspergillus fumigatus. And so these fungal adaptations to a climate-induced environmental change are going to indirectly and directly drive resistance to our current antifungals. And it actually gets a little bit worse. So there are 2 new classes of human antifungals that are in clinical trials right now. These molecules look great. We're excited about them. But they also are now being used in agriculture. So we're going to run into the same exact problem that we have with the azoles with these new classes of drugs that are being developed because they're being used environmentally, which is going to drive a resistance to these new molecules. So here's azole-resistant aspergillus. This is a high priority for the CDC now. A lot of data supporting this. It's estimated that 19% of aspergillus infections now in the hospital are resistant to the azoles. This is bad because this really is the drug that works. There's another one called amphotericin, but it's incredibly toxic to patients. It's difficult to treat patients with amphotericin. We now know that 7% of isolates in the United States are antimicrobial resistant, and we're finding these isolates more and more across the United States. The U.S. Geological Survey recently estimated that the agricultural use of azole fungicides is quadrupled in the last 3 years. So this is a real problem, and one that's only increasing with climate change and the use of agricultural azoles. So this is kind of where we're at, folks. We need to identify new mechanisms of fungal adaptation. to these different climate stresses to really fully understand their potential role in pathogenesis and virulence and drug susceptibility. We really need to focus drug discovery efforts with these fungi to target these biofilms that they form and they need to be able to be active in these changing environments, such as those that are deficient in oxygen, in order to overcome drug resistance and pathogenicity. We really want to be able to reduce this from happening. So we need to understand how climate change is changing the environment. So are there ways that we can prospectively, prevent some of these selective pressures from happening in specific environments, to prevent the emergence of new pathogens, to prevent the emergence of drug resistance? It's clearly much more than temperature that's changing. So we really need some perspective, environmental and clinical monitoring needed to catch these events as they're happening and to be able to deal with them. And so, just another little snippet of data from my lab is, we are developing new antifungals that are more active under low oxygen environments. Here's an example of one that we're currently developed. The data here is showing you that the molecule is more active against Aspergillus when oxygen levels are low than when oxygen levels are ambient. And so this is exciting for us. We have a molecule that may be active under environmental conditions that are being promoted by climate change. So I'll leave you with this witty quote, by Nobel laureate Joshua Lederberg. The future of humanity and microbes is likely to evolve as episodes of our wits versus their genes. And we're certainly facing now with climate change-induced genetic changes in fungi out in the environment. So I have a wonderful research lab here at Dartmouth. We have a wonderful research program at the medical school. We have a growing interest in climate and health at Dartmouth. We're happy to talk more about that further. And I'll be happy to take questions. And for some of the older members of the audience, you might recognize the Pillsbury Doughboy. So I'll leave you with that little joke. And with that, I'm happy to turn it over back to Paul.
Thanks, Rob. Wonderful presentation. A lot of information, very mind-blowing a little bit. But yeah, so we got some good questions in the chat, but I'm going to start by one just to bring up the analogy with the fungus that's been affecting the bat population.
Yes.
And if there becomes a antifungal, antimicrobial resistant fungus, what is the possibility that a similar fungus could do what human population has done to the bat population if we can't kill it? Is there a possible of a really pandemic fungus? We're not used to really pandemic fungi, but who knows?
Yeah, I think, the who knows is a good point. I mean, I think we have to be prepared. I mean, some of you on the call may have seen the TV series The Last of Us, where you have a fungal infection that takes over humanity. So people have thought about this. The biology of the organism suggests that, yes, it is possible. And I say that because of the bat incidents, the amphibians being wiped out. Fungi are the only organism known, other than us, to cause this extinction of another species. So that is documented. And so you have to say that, okay, if the right environmental conditions arose, the right pathogen arose, we could have a problem on our hands. Now, is that imminent? No, it is definitely not. I don't want anybody here leaving here thinking that that's imminent. But the azole-resistant isolates of Aspergillus are a huge problem in the clinic. So if you have an azole-resistant Aspergillus infection, your mortality rate is like approaching 90%. So, we are seeing these emergence of drug-resistant organisms that in the right patient population are lethal.
Okay, great. A question from the audience. Does the CDC do environmental surveillance for endemic fungi, or do they rely on case reports to create these maps?
Yeah, that's a great question. So the CDC mycotic branch is growing, for obvious reasons, I think, after my talk tonight. Hopefully you can appreciate that. Dr. Tom Childers, the head of that branch, they are trying very hard to set up a wider surveillance network, and they're having some success. They have a great system set up for Candida auris. Some of the endemic fungi, the only reportable one right now is Coccidioides mycosis. There are some states where blastomyces and histoplasmosa are reportable, but it's not national. And so, case reports are a lot of the, and medical records are a lot of the indications. The Vermont story that I shared with you tonight came from a search of insurance records because clinicians were starting to see it. They were talking, someone finally contacted the CDC and said, hey, I think something's going on here. And they were able to go back through the medical records and be like, oh yeah, there's definitely something going on here. So yes, Tom and his colleagues at the CDC are pushing hard now for more monitoring, more reporting being required of these infections.
Next question, is there evidence that the incidence of allergies or asthma from mold is on the rise? I'll just take a stab and I would say yes, clinically, that with warmer, wetter climates, you're going to have more mold growth and more allergies and asthma. But any more comments about that from your perspective?
Yeah, that's 100% correct. There's a lot of good data in the literature on this. The changes to our home, our environments, the environments we live in, where we live. After hurricanes, there are massive fungal blooms in people's homes. And, Hurricane Katrina, there was a lot of work done on fungi and increased incidence of allergy and asthma following Katrina. This is going to happen, yes.
On a personal note, last year and this year were the first time. I've been living in New Hampshire for 38 years. Last year and this year, the first time I've had mold problems inside my house because of the humid, damp air, and I've had to commit to running dehumidifiers all the time.
Yep.
Okay. One more from the chat. Can you describe the link between increased use of fungicides for plants and how that impacts fungal resistance among human pathogens?
Can you repeat the first part of the question?
Can you describe the link between increased use of fungicides for plants and how that impacts fungal resistance among human pathogens?
By length, I guess I mean length of time.
The link. I'm sorry, the link.
Oh, the link. Yes. So yes, definitely. So the azoles that are being used in agriculture, they're different than the azoles that are used clinically, but they target the same protein in the fungus. So they target an enzyme. It's called CYP51A or ERG11A. It's a cytochrome P450. It's involved in the synthesis of ergosterol, that lipid that's a really important, it's a sterol that's important part of fungal membranes. And so when they spray azoles in the environment, it saturates the soil, and these fungi are out in the soil living, so they're being exposed to a molecule that it's inhibiting their growth or killing them. And so you're creating an environment that selects for mutations in the fungal population that is resistant to the azole. And so it really is there's some really great studies. Most of them have been done in Europe right now, linking, like, for example, the widespread use of azoles in the tulip industry in the Netherlands with a massive increase in azole-resistant aspergillus isolates, because aspergillus is on plant material too. That's where it's at. And so it's being directly exposed to these agriculturally used azoles, and then you get the emergence of resistance. It's in the population, and
Right.
it kind of becomes a probability. Are you going to get an azole resistant isolate, or are you going to get a susceptible isolate?
Right. Well, what a fascinating hour. Just to remind people, Dr. Cramer has allowed us to record this evening's presentation. It's available on our website. Spread the word to friends and colleagues. Fascinating talk available for free from our website. So spread the word, check it out. Thanks so much.
Thank you, Paul.
So
Thanks,
much.
everybody.
And I'll be in touch tomorrow with the meeting link as well, so you can find it there.
Great.
Thank you so much. Have a good night, everyone.
Thank you all.
Good night.